Hello students, today's class is on cells of the nervous system. This is a virtual practical class during the pandemic time. Please imagine that you are in the histology lab of North Delhi Municipal Medical College and Hindura Hospital, and I am Dr. T.S. Roy, and the other teachers of the department are showing you the slides. The objectives of the class are that you should know how to study the neurons, glial cells and the nerve fibers. You should also know the how to identify a neuron, the supporting cells of the spinal cord, sensory and the autonomic ganglia. And we should study the neurons and glial cells of the sensory cortex, motor cortex and the cerebellar cortex. We will also study the microscopic features of the nerve fibers. Basically, we are going to study the neurons, the supporting cells, and the nerve fibers of the nervous system. How to study the neuronal morphology? Till now, most of the classes we have studied the cells by hematoxylin and eosin staining. Neurons can be stained with hematoxylin and eosin stain, but certain components of the neurons do not take the hematoxylin or eosin stain. Especially the fibers do not take this hematoxylin and eosin stain. So for that we use special stains. You already know what is the Nissl substance. The Nissl substances are the rough endoplasmic reticulum. They can be stained with special staining. Those are called Nissl stains. Those Nissl stains are thionin stain, cressyl violet stain, toluidine blue stain. They are basophilic stain. That's why they stain the nucleolus also and the Nissl substance and also nucleus of the neuroglial cells. In some of the places you can see the capillaries lined by the endothelial cells. The nucleus of the endothelial cells takes the Nissl stain also because they are the basophilic stains. What are the other methods of staining the neurons? There are H&E staining, phosphotungstic acid hematoxylin staining, silver impregnation. The silver staining is done to see the dendrites and the axons. Golgi method. And osmium tetroxide staining, it stains the myelin. This is a section of the spinal cord. What you see is the butterfly shaped gray matter and surrounding is the white matter. This is the dorsal part of the white matter and this is the ventral part of the white matter. The gray matter has ventral horn and dorsal horn, and in the center there is a central canal. The two halves of the spinal cord are separated ventrally by the ventral fissure and dorsally by the dorsal sulcus. The dorsal root ganglia, which is outside the spinal cord, this is a sensory ganglia, can be seen, and spinal cord is surrounded by the pia mater. In the higher magnification, you can see the central canal is lined by ependymal cells and surrounding which lies the fibers and the cells of the spinal cord. This is in hematoxylin eosin staining. This is the higher magnification of the ventral horn. The ventral horn of the spinal cord contains motor neurons. This is the ventral horn where you can see an arrow, and the bigger arrow shows the position of a neuron. In the higher magnification, the neuron is visualized, and see the central position of the nucleus, and the processes are coming out. Normally the processes do not take hematoxylin and eosin stain, so it is totally unstained areas. This is a more or less triangular neuron, and multipolar neurons can be seen also. And inside the cytoplasm, the Nissl substances has taken the eosin stain, but the Nissl substances are not so prominent as we have seen in thymine staining. And the small nuclei, what is visible, these are the nuclei of the glial cells. Already you know that there are three types of glial cells: astrocytes, oligodendrocytes, and the microglial cells. And with this stain, it is not possible to identify the individual glial cells. The visualization of the dendrites, axons are done by some special stains. These are called Golgi stain and the silver staining. In this staining, the silver nitrate is deposited on the fibers, either the dendrites or the axons. So, in this stain, it is possible to visualize the minute branches of the dendrites and axons. This is Golgi staining, rapid Golgi, combined with osmium tetroxide and potassium dichromate. The sensory and motor cortex. This picture shows the cerebral cortex, the superlateral surface of the cerebral cortex, and there are elevations and depressions. The elevations are called gyri and the depressions are called the sulci. This is the central sulcus. In front of the central sulcus is the precentral gyri, and that is the motor cortex, or if we take the section of the brain at this, we will see the motor neurons, and this controls the motor activities of our body. Just behind the central sulcus is the sensory cortex. The general sensations from the body, pain, touch, temperature, are relayed here. The ganglion cells or the stellate cells are more in the sensory cortex, and pyramidal cells are more in the motor cortex. This picture depicts the 6 layers of the motor cortex. The cerebral cortex, if you take a section, you can identify 6 layers: the molecular layer, outer granular layer, outer pyramidal layer, inner granular layer, inner pyramidal layer, and the multiform cell layer. In the motor cortex, this inner pyramidal layer contains the motor neurons. These are either pyramidal in shape, or in section it looks like triangle. The dendrites come out from the apex, and the axons come out from the base. This granule layer are the cells which is present in abundant number in the sensory cortex. In the sensory cortex, these motor neurons or the pyramidal neurons are less in number, and these granule cells are more in number. In the motor cortex, the pyramidal cells are more in number and the sensory or the granular cells are less in number. This is the hematoxylin eosin stent picture of the motor cortex, where one can identify the pyramidal neuron in a lower magnification. So these are the pyramidal neurons. Apical dendrites has not been stained. The basal dendrites can be seen. This is another one where you can identify some part of the apical dendrite, and this is in the layer 5 of the cortex. These white spaces are the blood vessels, and small nuclei are the nuclei of the glial cells. These areas which contains the glial cells, fibers and the blood vessels are called neuropil. This is in higher magnification of the sensory cortex. In low power, one can identify small granule type of or granular cells. There are some large granular cells. In higher magnification, you can identify this large granular cells and small granular cells. In the large granular cells, you can identify the nucleus and some amount of cytoplasm with Nissl granules. But in the small cells, cytoplasm cannot be identified. So, identify features of neurons are nucleus and the cytoplasm containing the Nissl granules. In all other places, we have seen the neuronal cytoplasm containing Nissl granules. So these are the neurons, these are the sensory neurons. This is a section of the sensory cortex, and the outer and inner granular layer of the visual cortex, auditory cortex or the general sensory cortex contains this type of neurons, the glial cells, and these empty spaces are the blood vessels. Individual glial cells cannot be identified with this hematoxylin and eosin stain. The nerve fibers has taken the stain, and you can see some white areas around the nerve fibers. Those are the myelin. Myelin doesn't take hematoxylin or eosin stain. So this is called the neuropil. The cerebellum, just like the cerebral cortex, the cerebellum has certain elevations and depressions. Those are called cerebellar. These elevations are called cerebellar folia. So cerebellum also has gray matter and white matter, and a section of cerebellum shows the individual folias are there, and outside one can see the different layers of the cerebellum. There are darker areas and lighter areas. In higher magnification, one can identify the different layers. So the darker area is the gray matter, and from the gray matter the axons come out and it forms the white matter. The whole area or this folia is just like cerebral cortex. The cerebellum has three layers: the outer molecular layer, Purkinje cell layer and the granule cell layer. So this is the gray matter of the cerebellum, and white matter of the cerebellum is here. What you can see where the pointer is moving, this is the white matter of the cerebellum. So this is the gray matter, and white matter is here, the nerve fibers from the cerebellar cortex passes through the white matter. This is the higher power of the cerebellum showing the gray matter. See, in lower magnification also one distinct big cell layer is visible. This is called the Purkinje cell layer. cerebellum contains special type of cells. Those are called the Purkinje cells. So these three layers, molecular layer, the granular layer and the Purkinje cell layer forms the grey matter, and the deep part is the white matter. This is showing the Purkinje cells. These arrows indicate the Purkinje cells, and this thick layer is the granule cell layer. In higher magnification, one can see the Purkinje cells, the central nucleus, the dendrites of the Purkinje neurons, and the nuclei of the granule cells. So three layers are there. The molecular layer, which contains the dendrites and some small cells, the Purkinje layer, the second layer, and the third layer is the granule cell layer, deep to which lies the white matter. As I was mentioning about the dendrites of the Purkinje cells, see the vesicular bodies of the or the soma of the neuron, and it has a bigger dendritic tree, just like the branching of the banyan tree or some other trees. These Purkinje neurons are very big neurons, and one can see the nasal granules, and these arrows indicate that it carries impulses from the periphery to the center. The axons carries these impulses away from the Purkinje cells, and this is a oligodendrocyte which myelinates the axons. This is in Golgi staining, the drawing of a dendritic tree of the Purkinje neurons. Now we'll study the sensory ganglion or the dorsal root ganglion, trigeminal ganglion. These are the sensory neurons, sensory ganglia, which are the dorsal root ganglia is present just around the spinal cord. You have already seen the dorsal root ganglia. The dorsal root ganglia sections, or if we take, it will show neurons, big neurons. See, these neurons, they have central nucleus, and you can identify the nucleolus also in the higher power. Surrounding the neurons, there are small cells, just like the glial cells in the dorsal root ganglia. These are called satellite cells. So the dorsal root ganglia has bigger neurons with nasal substances. This is a nasal stem, so it has coarse nasal granules. You can identify the nasal granules in the cytoplasm. Here is the nucleus containing the nucleoli. This is a typical picture of the dorsal root ganglion, which is circular in shape, containing the central nucleus. You know that dorsal root ganglia cells are pseudo unipolar cells. So the dendrite comes from one pole. That is how, if you take a section of the neuron, it looks round. And the surrounding cells are the satellite cells or the supporting cells. The nerve fibers or the axons come out, and they lie in between the neurons. So here the nerve fibers are present. Notice that groups of neurons are there, and they are separated by nerve fibers. Here mainly you can see the neurons and the supporting cells, and in the periphery you can see the nerve fibers. This is hematoxyl eosin stem, dorsal root ganglia, central nucleus, and Nissil granules, already we have mentioned that it doesn't extend, or it extends very mildly with somatoxylin. Here you can see the satellite cells. These are special types of cells surrounding the dorsal root ganglia or the autonomic ganglia also. In trigeminal ganglia also, the similar kind of neurons are visible. This is a picture of the autonomic ganglia. You have seen the sympathetic trunk during the dissection of the thorax. It lies by the side of the vertebra. So if you take a section of this ganglias, these autonomic ganglias are, there may be sympathetic ganglia or parasympathetic ganglia. The parasympathetic ganglia you have not seen till now. During the dissection of the head, face, neck, you will see the parasympathetic ganglia. So the difference between the sensory ganglia and the autonomic ganglia, you can see that neurons are there. Neurons in low power, it looks round, but see the position of the nucleus. In higher magnification, see the position of the nucleus. It is eccentric in position, away from the center. That's why it is eccentric. And the dendrites come out from different surfaces of the neuron. So they look irregular in cross section. See, this is the appearance. It is irregular in appearance. Although some of the cells are looking slightly oval, but most of the neurons are irregular in shape. So identifying features of the autonomic ganglia are the irregular cells, the position of the nucleus is eccentric, the Nissil granules are there, finer Nissil granules, and the satellite cells. See, the satellite cells are less in number in comparison to the sensory ganglia. Now, see the nerve fibers. The nerve fibers separate individual cells, but in case of the sensory ganglia, we have seen groups of cells are separated by the individual cells, because here the cells are multipolar. So the nerve fibers has to pass from the soma, and individual neurons are separated by nerve fibers. These are the blood vessels. Now we'll see, this is showing the neurons and the position of the nucleus. Now we'll see the difference between the sensory and motor ganglia, sensory and autonomic, sorry. The shape in case of the sensory ganglia is round, but in case of the autonomic ganglia, it is oval or irregular. Already I have explained why it is irregular, because they are multipolar neurons, and the sensory ganglia is unipolar or pseudo-unipolar neuron. The position of the nucleus is most important. In case of the sensory ganglia, the nucleus is central, and in case of the autonomic ganglia, the nucleus is eccentric. The satellite cells are more in number in case of the sensory ganglion. The satellite cell function is to give support, the metabolic support, physical support. As these neurons are very large, they need more of satellite cells for their metabolic, for their metabolism. And in the case of the sensory cells, the number of satellite cells are less. And arrangement, already I have explained. The sensory neurons are arranged in groups separated by nerve fibers. And autonomic neurons, the individual neurons are separated by nerve fibers, not in groups. They are not separated, or they are not arranged in groups. The supporting cells of the central nervous system, or the cerebral cortex and cerebellar cortex. Already we have identified under the nasal stem some small nuclei without the cytoplasm. Those are the glial cells. The glial cells can be studied with some special stain. They contain some protein called glial glial fibrillary acidic protein GFAP. If it is stained by some antibodies, then the fibers can be seen. Their fibers can be seen. So there are two types of astrocytes one can see, the protoplasmic astrocytes and fibrous astrocytes. This is the protoplasmic astrocyte in the gray matter of the cerebral cortex, stained by glial fibrillary acidic protein, and the fibrous astrocytes are present in the white matter. The largest white matter is corpus callosum. So these are the fibers of the fibrous astrocytes, the stained areas. The unstained areas are the neurons. As they support the neurons, that's why they are present around the neurons. But more fibrous astrocytes are present in the white matter. Similarly, oligodendrocytes and microglia are stained with some antibodies. Here, see the oligodendrocytes and the microglia. The microglial processes are very small. It looks like a spider with their legs. Now we come to see the section of the nerve fibers under hematoxylin and eosin stem. A nerve fiber is covered by fibrous tissue that is called the epinurium. The epinurium sends connective tissue and it divides the nerve fibers into individual groups. Those are called perineurium, and blood vessels pass from the epinurium to the perineurium, and then the perineurium also sends connective tissue between the individual axons, that is the endoneurium. So this shows a perineurium and the bigger arrow shows epinurium. In higher pictures, higher magnification, the endoneurium is visible. BNF is the bundle of nerve fibers. This is in hematoxylin and eosin stem. The hematoxylin or eosin do not stain the myelin, that's why it looks white. The axon. So this is the group of nerve fibers, bundle of nerve fibers, and inside this is the axon. Individual axons can be seen, and endoneurium is the connective tissue which is stained, which has taken the eosin stain. These are the axons, this is the capillary, and these are the nucleus of the Schwann cells which myelinates the axons. So you know that myelination of the axons in the peripheral nervous system or in the nerves are done by the Schwann cells, and myelination of the central nervous system is done by the oligodendrocytes. So this is the connective tissue, this is called the epinurium, and this connective tissue going inside the nerve fiber, this is called the perineurium, which carries the blood vessels for supplying the axons and the other materials inside the nerve fiber. This is the longitudinal section of the peripheral nerve showing the perineurium. This is in the lower power. The perineurium is present here. This is the cross section in higher power. The axon is seen here, and these are the myelin unstained portion of the myelin. This is the Schwann cell nucleus. This is the Schwann cell nucleus, and here is the endoneurium. This is the nucleus of the fibrocyte or the fibroblast also. This slide shows the transverse and the longitudinal section of a nerve with myelin stain, that is the osmium tetroxide stain. The osmium tetroxide stains the myelin black. One can see the nerve vesicle, and inside the nerve vesicle the darker areas are the myelin around the axon. The connective tissue also takes some amount of stain. So this is the perineurium, and outside is the epinurium. The endoneurium will be visible in higher magnification. Here, this is the perineurium, and the whole structure shows the myelin. This is the electron microscopic picture of a nerve fiber showing the myelin sheath with the alternate dark and light uh, lines. This is the major and minor dense lines in the myelin sheath. Inside is the exoplasm containing the mitochondria, neurofilaments, neurofibrils, and these are the Schwann cell cytoplasm, outside which lies the connective tissue or the endoneurium. So, in this class, we have studied the nerve cells, the supporting cells, the nerve fibers, and we have talked about some of the stains used for identifying the nerve cells. Please see or please read from a textbook and see the pictures. If there is any doubt, please call your teacher for doubt clearance.